If you've spent any time in a genetics lab, looking at you several times, then you've had to go through the process of extracting DNA. When I was in university, the only way they taught you to do this is with a silica column. The idea behind these columns is that DNA is negatively charged, as is silica. Under neutral conditions or in distilled water, the two repel each other, but when you add a special salt, the positive ions form a bridge between the DNA and silica, making them stick together. To extract DNA using this method, you first break open the cells, then add your salt mixture to your sample. Then by loading the solution into a column and running it through a centrifuge, only the DNA will stick and all of the rest of the cellular junk will pass right through. Then you can switch out the collection tube that sits underneath with a clean one, and run distilled water through it to make the DNA let go and collect your product. The problem with this is that the whole process is rather tedious and can take up to an hour and a half between all the waiting and spinning. Sure, practice can bring that time down, but it could still be better. So one day while I was sitting in class, I got to thinking, what would happen if you put the silica on something magnetic instead? Then instead of running a column, you could just break open the cells, drop in the particles in salt, and then use a magnet to pull them out and drop them into clean water, causing the DNA to drop off. I didn't know at the time that groups like Thermo Fisher and other biomedical supply companies were working on the same idea, so I set out to see if something like that could be made. Turns out it's actually incredibly easy, and I'll be taking you through my method for preparing these sorts of systems. Before we begin, I will note that there's potential for collaboration that I'll cover at the end of the video, so stay tuned for that. All we need for this synthesis are the following: any soluble iron 3 salt, any soluble iron 2 salt, some ammonium hydroxide, and some sodium silicate. The higher purity everything is, the better this is going to work, but here I'm just using off-the-shelf reagents. For my iron 3 salt, I'm using ferric chloride etchant, the same stuff I used in my PCB making video. For my iron 2 salt, I'm using ferrous sulfate, which you can buy at most garden stores. My ammonia is 30% clear with no perfumes, and my sodium silicate comes as a 36% solution in water. First, we'll be making the magnetic core of the system using the co-precipitation method. To do this, we mix our iron salts in a ratio of 2 to 1 iron 3 to iron 2. With my starting materials, this works out to 7 milliliters of the etchant and 2.5 grams of ferrous sulfate. Dilute this to 40 milliliters with distilled or deionized water. Then we set our reaction on a stir plate, add a stir bar, and set up a burette or SEP funnel above so that we can drip in the ammonia slowly. We'll need to add about 140 milliliters of ammonia, or until the solution hits a pH of 10. The slower you drip in the ammonia, the better the particles will be to a point, so the total duration should be about an hour. As the ammonia is dripped in, the solution will start to change color, first to an orange-brown, but will get steadily more black as the pH rises. The precipitate won't be magnetic until you hit pH 10, so don't be surprised if the particles don't respond to a magnet at first. When all's said and done, we're left with a large amount of magnetite particles that are very small. Because of this, we can't really filter them out using most filters, but because they're magnetic, we can clean them another way. Use a pipette to draw up some of the black precipitate and put it in a test tube. Add a fair bit of water and mix well. Then use a magnet to collect all the particles at the bottom, and pour off the top solution. Repeat 3 to 4 times to clean off any remaining salts and ammonia. With our cores done, all that's left is to coat them in silica. There's a variety of ways to do this, and I suspect that Thermo Fisher and other companies either use something like tetraethyl orthosilicate or some other silane. Instead of that, we'll be using sodium silicate because it's both the easiest and cheapest soluble form of silica to get a hold of, and can be purchased online or at many hardware stores. In future videos, we'll be exploring what the other silanes can do, as many of them leave useful functional groups on the outside of the silica layer that other molecules can be attached to. A great example of this is the NHS fluorescein that we prepared in a previous video. Resuspend the particles that we just prepared in some water and transfer them to a Teflon hydrothermal reactor. Here I'm using a 100 milliliter reactor. Then add 1 milliliter of stock sodium silicate solution to the reactor before filling the majority of the rest of the chamber with water. Assemble the reactor and place it in an oven at 180 degrees Celsius for 8 to 10 hours. When the reaction is over, this is what we're left with: magnetic particles covered in a very thin layer of silica. Since we only use a small fraction of the magnetite we prepared, we can repeat this as many times as we like to prepare massive amounts of particles. Thermo Fisher's kits range from $200 to $1,000 per kit, which lets you run 500 to 100 reactions. What I like about my method is that it's possible to produce enough particles to run thousands of samples for only a few dollars if you've already got the chemistry equipment lying around. To be clear, I'm not saying that these will ever be as good as the professionally made stuff, but for the garage scientist on a budget or teacher in a classroom, I think these could be great. Before I can even consider that, though, these need to be tested more. And who knows, maybe these will be comparable and making them will become standard for citizen scientists. But there's still the question of effectiveness, and this is where I hit a bit of a roadblock. When I was first testing this, I had access to my professor's lab, and most importantly, his Nanodrop spectrophotometer, which allows you to precisely measure how much DNA is in a solution. My first test showed promise, and I was able to successfully pass DNA between vials by changing the salt concentration and moving the beads around. I only managed to get in a couple of tests before I left school to work on other projects, and now I no longer have access to a Nanodrop and can't do any more testing. So this is where I need your help. If you've got access to a Nanodrop or Qubit fluorometer and some DNA, send me a message at thethoughtemporium@gmail.com. I'll ship a batch of these to you, along with the protocol so you know how to use them. I'll also include some of the salts I used in my original protocol, but I would like to also encourage you to experiment and see if other salts or combinations of salts work better. Be sure to take detailed notes, as I want to turn this whole procedure into a paper and publish it in an open access journal. Anyone who helps with this project will be listed as a co-author on the paper. Thanks in advance for all of your help and continued support on all of my projects. I'll post updates on the project as things progress, and will be sure to share the paper when it's eventually written, so be sure to check back every other Monday for all of that. If you enjoyed the video, be sure to click the subscribe button and leave a rating. If you want to support the continued production of videos and research like this, consider heading to my Patreon, or have a look at the merch in my store, like this awesome quantum mechanics poster. That's all for now, and I'll see you next time.